Hello guys, today we're going to be talking about the required practical activity 1, and we'll be able to observe, draw and label a selection of plant and animal cells. A magnification scale must be included. Okay, so what does this mean? So when you do your required practical, you must include this magnification scale, and I'll go through the steps of how you should go about it when you're doing the practical. This bit here is telling you about the estimation and explain why when they should be used to judge the relative size and area of a subcellular structure. So basically you should know the generic size of, is it micrometers, is it millimeters, so what sort of sizes are we expecting of the different sort of things we should see in the cells, having a good idea of that. So that's the sort of thing they're expecting in this. And this bit here is just when you go to draw your drawings, you need to be able to, like, the teacher will be showing you comparisons of how does your drawing look to someone else's drawing of the same thing, and you have to be able to have the ability to draw it yourself, the thing you see actually. So going to the actual required practical, let's look into it in more detail. So we have a few things. Firstly, we need to learn a little bit about the light microscope. This will be further developed in the microscopy video. We have to observe, we have to draw, and we have to label a selection of a plant and animal cell, and magnification scale must be included. So let's just have a quick understanding of what a light microscope does. As I said, I will go through it more in the microscopy video as well. So what does the light microscope actually do? So the way it works is that we have this eyepiece, right? So the eyepiece is going to be the place where our eyes actually see the the actual cell. So from this bit we have also what we call the objective lens as well here. Okay, so there are two different lenses, and what they'll do is this will go to the cell up here, to the eyes up here. So it goes up to the eye up here, and then on the stage itself you will have the specimen. The specimen, the light rays will shine right through, okay, and then obviously if it's opaque, when you see a cell, it sort of looks like this, and you have, like, let's say shaded areas here, which is the nucleus here, and there's a cell membrane. You'll see this because these structures here are opaque, okay, so that means that they don't allow light through. But the structures in between are usually translucent, which means they can let some light through, or transparent, which means it allows all light through. And the nucleus itself is it's semi-translucent, and some are opaque structures in this structure. So yeah, so light goes through and it comes to the eye. Our eyes then detect it and can actually see it. So what are the things that we need to know from the actual microscope equipment we're going to use? So we need to know that the fact that there is a light source that we need. It can be battery powered, or it can be powered through the directly by the power socket. We have a few adjustments that we need to make. So fine adjustments and coarse adjustments are slightly different. So coarse adjustments are making big adjustments. So this stage makes really big movements. Fine adjustment means it makes very, very, very small adjustments from the stage. Okay, so this bit is called a stage because it's the stage for the slide that's actually going to go here, right? This bit here is going to be where your slide sits, and you're going to clip that slide on. Right? You're going to so when you have the slide, these are the clips. You're going to put that clip underneath the slide, so it just clips on top, and they're going to stay like that. And then you'll have whatever specimen in the middle here. So that will allow you to see it. So let's see, the first thing we need to do before we get onto our microscope again is we need to be able to prepare the slide. So I'm going to do it for an onion specifically because that one's easy to explain. So the first thing we're going to do is add a drop of water to the middle of a clean slide, and that's going to allow us to actually get that onion to stick on, alright? Because if it's very dry, let's say you have a very, very dry piece of leaf or something, it's not going to stick onto the slide. You need to have some water or some sort of adhesive to put it on, and water is a good one for this one here. they may ask you a question as why white water be bad to put in, and that's because it depends on the type of water you put in. So why can water be harmful to your specimen? So water, it has osmotic properties. So that means that water can go inside the cells, right? So if it's an animal cell, the cell will burst, right? But we don't have to worry about this for a plant cell. Why is that? Because let's go back to this previous spec when we learned in 4.1.1.1, we learned that plant cells have what? A cell wall. Okay, so they have a cell wall, and that's the important thing. Because it's a cell wall, it won't explode. Okay, it won't explode, or it will rupture the cell membrane. So that's why water is okay to use here. Then we cut up the onion and separate into layers, and we choose it to peel off some of the epidermal, which just means from the surface. So basically the surface tissue from the bottom of one of the layers. Okay, so why is it that we're going to use epidermal tissue, and why is it that we take it into layers? So the layers is used because we want a very thin layer to allows light through. That is important. They could ask this kind of question in the exam, to allow light through. We need to use the tweezers to place the epidermal tissue into the water on the slide. Now, why would we not want to use our fingers? That's a reasonable question. If you use our fingers, there's dirt on our fingers, so that could contaminate our specimen. Then we have to add a drop of iodine solution. Now, this is a very, very common question they'll ask in the exam. And the use of iodine solution is to add contrast. What does contrast mean? That means it is able to distinguish very fine structures. So dyes, like solution like this, iodine solution, can stain a very specific substance or a thing. So something like iodine stains starch, and so you see any sort of starch like things or starch granules in the cell very well. Okay, so things like chloroplast should be very, very vivid with an iodine solution, and you will see the cell membranes and things like that. And then you have to place a cover slip, and the cover slip has to be upright. And the thing about the cover slip you must mention, you have to tilt and lower it carefully to cover the specimen, and make sure you don't get any air bubbles in. That's a big point here, because if you get air bubbles in, what happens? You can't see the specimen properly. It's what we call an artifact. Or you get a bubble is what we call an artifact. Okay, so artifacts are anything that can obscure our view from seeing the actual specimen. So if you want to actually look at the slide, what do we do? As I said, we have to clip it onto the slide, right? So onto the stage, sorry. So we clip the slide onto the stage, going back up. This is the stage. We clip it on, and then we put it to the lowest magnification first. So we want to see a general view first, and then we use coarse adjustment. Okay, we're not going to use fine adjustment. That's going to take way too long. All right, that's the reason why we don't use fine adjustment first, is because it takes too long. So coarse adjustment is the way to go first, and we're going to place it just below the objective lens. Why is that? We do not want to break so it stops breaking the slide. Because imagine you, let's say you start midway and you're moving the knob down, up and down, up and down, and you actually end up smashing the thing. So by doing this and you're putting it right below, the stage below, that means you know you're never going to put the stage going up. You'll always make the stage going down, right? So when you look down the eyepiece, you're going to move the stage down, right? Always down, or you're going to break the actual slide, and people do that a lot. And then you're going to adjust it when you see like a roughly that's in focus. You're going to adjust it with fine adjustments, okay, until you get a clear image. Then you're going to swap it to the higher magnification. Right. So higher magnification. Okay, that's a typo. Yeah, higher magnification. And then we're going to repeat steps 3 to 5 again. The way we use coarse adjustments again, and then focus down and find the image. Now, how do we draw this in observation? Okay, this came up for my A-level exams, so it could come up for your GCSEs as well. So do learn this, all right? What are the important points of drawing observation? The first thing is using a pencil, and make sure you use the sharpest point. I know these are very common sense points, comes up in your exams, so you best know it. Make sure takes up half the space. Your teacher probably has mentioned this tons and tons of times. Do not draw a small diagram. Mention that in the exam. How do you draw? What's the way to do a perfect observation? It has to take up half the space of the piece of paper or whatever that you're trying to draw on, and it has to be very clear without broken lines. So it has to be a continual diagram. And the drawing should not have any coloring or shading. Okay, that's important, right? Some teachers tell you that you can shade it after. No, during observation you're not shading or coloring. Okay, that's not an observation at that point. Subcellular structures should be in proportion. So what does that mean? So if we look at this structure here, we can see the nucleus here. If I was to draw a ribosome, I can't draw a ribosome this big. That's not a ribosome. Okay, ribosomes are small. So you're going to draw like dots like this, right? These are sort of ribosomes. Right? It's much smaller than the nucleus, and you have to put it in proportion. It doesn't have to be exactly the same size proportions. That would be ideal, but that would be very difficult to do. But you have to get a reasonable proportion. So if you see very, very small, draw them very, very small. And then you must include a title and a magnification. This magnification is mentioned in the specification. You must mention it, or you'll lose a mark if you don't. And then label any important features. So usually in cells and on your cell, you usually see the nucleus. You'll definitely see one. And you'll definitely see the cell membrane. So you can label these two easily. Ribosomes may be difficult to see. You might be able to see the chloroplast if you're lucky, etc. So moving forward, the equation that you need to be aware of is that magnification is equal to image size over the actual size. Just remember this triangle to working things out. I'll go through this more in the microscopy video about the calculations and how to do that, but that's not the main focus of the video today. There is this also. This point is quite a big focus. I will explain more about what micrometism things mean, but I'll explain that now because it is part of the spec. So what this means is you need to remember that when you talking about cells, they are very small. They're not millimeters. Okay, this is not millimeters. This is micrometer. Okay, so they're not milli. So this is 10 times to the 10 to the minus 3 times smaller of a micrometer. So that means, so what does that mean? That means it is 1,000 times smaller than a millimeter. That's how small a micrometer is. And it's 50 micrometers. That's very, very small. A cell membrane is even smaller. It's 7 nanometers, right? That's another, that's 1,000 times smaller than a micrometer. Okay, as I said, we'll go through this in more detail in the microscopy video, but yeah, this is very important. Knowing these sort of values, I would say learn a few of these values, okay? And I think it's quite important to learn some of these values for the exams, to have an idea as to, oh, so this is about this around the size. You're not going to be expected to know the exact value, but you need to know a rough value. So I'd say if you learn that nucleus cells, that's 550 micrometers, and you learn the nucleus around 10 micrometers, you'll be fine. You won't lose marks. When you do your past papers, you'll find out what sort of numbers they sort of expect, but AQA can be a bit fussy and they can change the mark scheme day to day. But if you are in the right region, you should be able to get the marks. So that's all for me for the required practical, and that's all you need to know. And I hope you've enjoyed the video. Thank you.